Okay, I'm going to be talking a little bit about the overview of animal diversity. This is a very, very short chapter. I'm just going to talk a little bit about what does it take to be an animal, show you the family tree. It's kind of like the roadmap of where we're going to go in this semester. And then talk about just some basic body plans that some animals have and just the origins of animals, really. So, general features of animals is that obviously they're very diverse, but there are some very important characteristics that they do share. They are heterotrophs, which means that they have to consume other organisms. They are multicellular. They don't have cell walls. They have cells that are specialized that carry out a specific job. They're able to move, most of them anyways. I'll talk about some exceptions on that one. But movement obviously means you have some type of musculature and nervous system coordinating together, very diverse in form and habitat. They reproduce sexually. And then their embryonic developments can also have or be unique. So we'll talk a little bit about deuterostomes and protostomes here. Now in your book, this would be chapter 33 in your books with the new edition. Okay. So here's kind of the roadmap where we're going to go. And we have a question mark down here, the Choana flagellata. Remember, we believe these guys were kind of like the early ancestors or where animals might have branched off from. But, you know, when we start getting into animals, we're going to talk about the parazoas first, sponges, okay? And parazoa means near to or next to an animal. It doesn't have all the unique features of an animal, but it does have some specialized cells that protists lack. Eumetazoa means true animal. And then from there, we're going to talk about like symmetry. Radial symmetry, we'll talk about the ctenophora and the cnidarians, and then the rest will be bilateral. And then with bilateral symmetry, means now we'll get into the deuterostomes and versus the protostomes. And so we'll talk about each of these little clades. There's chordata, which basically means like backbone or vertebrae, and then protozoa or protostomes over here. So we have a lot of different clades that we're going to go through in this semester. Okay, so the evolution of the body plan, basically 5 key transitions. First off, evolution of tissues. Okay, and this allows for that specialization to occur. Because, I mean, like if you just have one cell, that cell carries out all the functions for that organism if it's unicellular. But now we are going to be multicellular, which means we have made of many cells, and each cell might, you know, carry out a specific job. And so the simplest animals are the parazoas, which are sponges. They lack tissues and organs, but they do have cell specialization that's not found in protists. They have the ability to differentiate as well as de-differentiate. So we'll talk a little bit about that. As well as to disaggregate and aggregate their cells, separate from one another, and gather back together again. So when we talk about the parazoans, the sponges in depth, we'll definitely cover that. But all the other animals, the eumetazoas, the true animals, do have distinct and well-defined tissues. And differentiation, basically the cell fate, is irreversible. Once it's on that pathway, that's the cell that, or the type of cell it's going to become. Second kind of key transition for the animal body plan is most animals exhibit radial or bilateral symmetry. So radial symmetry evolved in our marine organisms, the phylum Cnidaria, as well as the Ctenotophoria. And you guys know what radial symmetry is. You can cut it any way and you're going to have symmetry. Okay. Now the other type is bilateral, which means you can pretty much only cut it maybe 1 or 2 ways. But you get a left half and a right half, and they're mirror images of each other. And with bilateral symmetry, one thing I want to point out is cephalization. Okay, so we have a dorsal side and a ventral side, so a top and a bottom. We have a front and a back. But now we have a specialized area on the organism where there's a lot of sensory organs. And it might help them, you know, find food or move more efficiently than radial symmetry. So we'll discuss a little bit about cephalization when we get to bilateral symmetry. Third key transition is body cavities. Okay, body cavities were, you know, allowed us to have advanced organ systems. And so with embryos, when an embryo is developing, we basically have 3 germ layers: an ectoderm, an endoderm, and a mesoderm. And the ectoderm means like outside, so think skin. Okay, It is also responsible for the nervous systems of organisms. The endoderm is like inside, so digestive and intestines, and the mesoderm is going to be kind of like in the middle, your skeleton and your muscles. Okay, so we're going to talk about different body cavities, like the pseudocoelomates and the coelomates and I'm missing one, I can't think of it. I think there's like 3 of them, acoelomates. So we'll talk about different body cavities, I think, in the next chapter. Oh, there it is, right here on the next page. So the acoelomates, no body cavity. So we'll give you some examples of that. The pseudocoelomates, it's not enclosed by a mesoderm. Basically, it's located between the mesoderm and the endoderm. And then we have a coelomates, which means that the body cavity is enclosed by a mesoderm. So here's a picture just showing the 3, acoelomate, pseudocoelomate, and the coelomate. Okay, so how do pseudocoelomates circulate materials? Like they use it by churning fluid within a body cavity. Coelomates cannot, and so that problem was solved by a circulatory system. And again, when we get into animals more in depth, we'll have an open circulatory. We'll talk about what does it mean to be an open circulatory and a closed circulatory. And in fact, right before, I believe, let me just check my calendar real quick, we will get to the coelomates next Thursday, right before Good Friday. And when you guys come back from Easter break, that following Thursday and Friday, we'll actually do a virtual earthworm dissection and talk about which type of circulatory system do earthworms have. Okay, but with an open circulatory system, blood is going to go into sinuses, it mixes with body fluids, and then it re-enters vessels. So it kind of pools in certain areas. Whereas in a closed circulatory system, blood is separate from body fluids. And so we have a closed circulatory system where all of our blood is always traveling in some type of vessel, basically. Okay, 4th key transition for animal body plan would be the developmental patterns. So I know I skipped embryonic development, because we were a little bit behind, but just, when a cell starts out, it divides, and it can divide very rapidly, and then it starts to, cells start to migrate and they start to shape the organism, if you will. And so we get these mitotic cell divisions of an egg to form a blastula, basically a hollow ball of cells. And then the blastula is going to become something called a blastopore, which means we get this indentation or primitive gut cavity. And the blastopore, it can either become the mouth or the anus of the organism. And so if the mouth develops before the anus, the blastopore, the fate of the blastopore, means that it's the mouth. So protostomes means the mouth forms before the anus. Okay, this includes our bilaterans, flatworms, nematodes, mollusks, annelids, and arthropods. And the deuterostomes means that the mouth develops after the anus, which means the blastopore will become the butt. So in chidoderms and chordates, and we belong to the chordates. So I don't know if your notes, if you just want to basically cut your paper down in half, because I'm going to talk about protostomes on the left-hand side and deuterostomes on the right-hand side here. But cleavage patterns do belong to this basic pattern. Basically, after the first couple rounds of mitotic division, you get this either symmetrical or asymmetrical division going on. So protostomes, well, if we take a look, they have spiral cleavage. So when they reach that 8-cell division, these 4 cells at the top are going to be asymmetrical in the division, and they're going to lean to one side, and you get this nice little spiral cleavage. Whereas with deuterostomes, they are directly above each other, okay, and you get radial cleavage. So diagram just showing protostomes in this 8-cell stage versus deuterostomes in that 8-cell stage, and you can kind of see that these 4 are not on top of the bottom 4. Okay, determinate versus indeterminate development. With protostomes, each embryonic cell is predetermined. Okay, it's already decided what it's going to be. But in deuterostomes, it's indeterminate. After the first couple rounds of cell division, it produces identical cells, and any one of these cells can develop into a complete organism. So that's another difference between protostomes and deuterostomes. Blastopore, which is the first little indentation, in protostomes, it becomes the mouth. In deuterostomes, it becomes the anus. The formation of the body cavity, in this case the coelom. The coelom will form spaces by the migration of mesoderm cells. And the deuterostome is formed by invagination of this archenteron or primitive gut. Okay. All right. Finally, the fifth transitional plan for animals is segmentation. A lot of animals do have segmentation, which means that systems are very redundant or copied, but it did allow for more efficient locomotion, which means movement. And so each segment goes on to develop a more or less complete set of adult organ systems. We call that the redundant system, which means that if any one of these parts gets damaged, it's not going to be fatal because they have lots of other redundant systems to pick up the slack. And then obviously locomotion is a little bit more efficient because segments can move independently and they're just a little bit more flexible in their movement. Okay, moving on to the traditional classification of animals. So animals, we usually classify it into 36 phyla, and I don't think those pages are accurate. They're really close, though. Let me just double check for you real quick. Nope, because that's still in fungi. I'll just keep paging through here just one more time. Yeah, I'm not finding it. I'll find it, though. But they are based on shared morphological and embryological differences. So that is one way that we classify animals. We look at their tissues and symmetries, and we break it down into main branches, where I just showed you, if I just escape out of here and go to this diagram right here. This is kind of the traditional way as to how we've classified animals. Parazoa to eumetazoa, going on to symmetries, and then going on to the embryonic development part. Let's see if I can remember where we were at. Okay. So symmetry and embryonic layers have helped us distinguish between diploblast and triploblast. Diploblast just means they have two tissue layers, an ectoderm and an inner endoderm, that forms during embryonic development. And so really, diploblasts are animals that just have radial symmetry, the kinetophores and the cnidarians. Triploblast means they have all three germ layers in the embryonic development, the ectoderm, the mesoderm, and the endoderm. And so this is basically animals with bilateral symmetry. So again, sorry, I'm just going to go back to here. Okay, these guys right here would be the diploblast, and the rest of these would be my triploblast. So there we go. Here's the diploblast. You only see two germ layers. The triploblast, you see all three. However, now with technology, we have a new look at the metazoan tree. Metazoan means true animal. And it is being reevaluated. There's been lots of case studies. We're looking at parasites of echinoderms and trying to figure out where did they belong. There are some that just don't fit with the clades at all. And so we've actually had to rely a lot on molecular data. And the findings, they kind of mess up our traditional ideas of animals. And I think the next chapter I actually show you the traditional and then the new molecular data tree. So I think I'm just going to wait on that to describe that tree. But anyways, it means that key morphological characters are not conservable characteristics that we had, thought that they were. Like a lot of times we just look at an organism and that's how we classify it, just by looking at it. But, that trait could have arose independently or in a different side of the animal tree. So molecular systematics, yeah, it's definitely changing how we classify animal phylogeny. So then basically, where did animals come from? So I'm going to talk about three hypotheses here. So the animal kingdom is monophyletic. Okay, parazoans, which are the sponges, and eumetazoans, which are true animals, do have a common ancestor. And so the question is, what is that common ancestor? If I go back to this family tree, notice it says Cyanoflagellata colony, and there's this question mark here. So what is that question mark? They're not exactly sure if that's true. So there are three hypotheses for the origins of metazoans. A multinucleate hypothesis, which just means that maybe they arose from multinuclear protists that look like today's ciliates. The second hypothesis is colonial flagellate hypothesis, which means we came from colonial protists, and a lot of it has to do with the chianal flagellate. That's supposed to be enough. Flagellate protists that resemble sponges. And the third is the polyphyletic origin, which means sponges evolved independently from eumetazoans. And of the three, colonial flagellate hypothesis is the favorite due to our RNA sequence data that kind of backs it up. But that's as far as we've gotten with that. So molecular analysis can maybe explain the Cambrian explosion. The Cambrian explosion occurred about 543 to 525 million years ago, and it was just this rapid explosion of diversity of animals where we can actually see nearly all of the body plans that we see today in the Cambrian period. And since the Cambrian period, we really haven't seen any new innovations in body plans. And so we just think, maybe that all that data that we're looking at with molecular analysis might explain why we see all these, this explosion during the Cambrian period. But I think that is it for the overview of animals. After this, we will jump into the non-coelomates, which I believe, actually, I'm going to have to look through the book because the book is designed a little bit differently, our new edition. But anyways, that is the overview of animals. I think the only thing you have to do is a one exit question card, or a one question exit card. And yeah.